Good morning or good afternoon, everyone. Today we have the pleasure to have Professor Hongtu Zhu from University of North Carolina at Chapel Hill as our webinar speaker. Hongtu is an internationally recognized expert in statistical learning, medical image analysis, precision medicine, biostatistics, artificial intelligence, and big data analysis. He has been a fellow, ASA fellow, and IMS fellow since 2011. He received an Established Investigator Award from Cancer Prevention Research Institute of Texas in 2016 and received the Inform's Daniel Wagner Prize for Excellence in Operation Research Practice in 2019. He has published more than 300 papers in top journals, including Nature Science, Cell, Nature Genetics, and also Statistics, JASA, JRSSB, as well as more than 50 conference papers in top conferences, including NeurIPS, AAAI, KDD, ICDM, MICCAI, and IPMI. Without further ado, let's welcome Hongtu.
Yeah, I think it's my great pleasure to be here, and it's quite my honor. And I think the talk the title of my talk is about the statistical learning methods for neuroimaging data analysis applications. And if you're interested, this is my website, you can visit it and download some of the materials. We usually put all my materials in our website. And this is basically, there are four parts of the talk. Basically, the first one is a review of neuroimaging techniques. The second part, neuroimaging processing analysis methods. The third one is large scale neuroimaging related studies. And the last one is population-based statistical analysis methods. And I just tried to review the neuroimaging techniques. These don't have to be neuroimaging actually. And this all the imaging techniques can be used for other organs. And like a heart or the liver, all these different different organs. And actually, in my review paper, I reviewed 8 relatively popular neuroimaging techniques. And it's basically, that include structural MRI and all the diffusion weighted MRI, function MRI, PET, CT, EEG, MEG, and the functional inferior to spectroscopy. And these are 8 popular ones. Each of the image modality has its own tracers, okay? And also have the data dimension, and also you track the features and the main and the clinical and research applications. And actually, there's many more of these kind of imaging technologies have been developed in the last 20 or even 30 years. And actually, you know, for most of the imaging data, I personally haven't able to touch it, and don't be afraid of analyzing this type of data. Every time is when I encounter new data type, I just go inside, figure out the details, and do all the things. And but for all these different imaging modalities, essentially they try to measure brain anatomy and also structure connections and the functional connections and the metabolisms. That's the different kinds of technologies about. And I just show some pictures about like the CTs. CT is super important. And it's in particular when you do the cancer imaging, CT is one of the major #1 kinds of imaging modalities. And related to brain is like larger blood vessels is very important for stroke type of these kinds of and neurodegenerative disease, in particular related to stroke. And also the PET, but the PET is a little bit invasive because you have the injector tracers and basically, and then allow these tracers to go to the different part of your body. And this is basically the EEG or the MEG stuff. That's another set of the methods people and basically use to measure the function for the function of brain functions and at the different brain regions. And this is another set of the imaging data. And basically in the brain, the most important part, and give you some basic ideas about basically in the brain, this is the gray matter and the white matters, okay? And also this is basically the mining sheet, basically the other speed of signal conductions. And axons plus the sheet, that's equal to neurofibers. There's a left part, you'll see these are neural structures. This is the dendrites, cell bodies. That's the basically is the white matter, the gray matter. And the white matter is this part, is axons. There's a nucleus, muscle mining sheet, and all these basic signal directions is basically transferred to your bodies, okay? And the major white matter by the pathways aggregate many fibers into bundles. That's basically how the brain looks like, okay? And for different organs, they have different structure and the functions. But actually, there's many things is shared. And this is basically is a function for my the MRI images and for the babies. And this is a different population of the brain. And you'll see this different regions and also white matters, okay? And this part is the function of my data, and also these are the white matter tracks, okay? And this is the right side of this, basically the cortical surface, and also. And in practice, when you have all these different imaging technologies, they try to use it, you know, scientists or physicians use this type of technologies to acquire the data. About here is about the brain. You can also do other organs. And actually, this is a very important, basically the multimodal approach these days is very popular, but it is easy to say, it's difficult to do. And the main reason is there's a lot of complexity behind it, but that's the kind of research people push it forward a lot, okay? And basically you have the different devices, that's the different models and the different scales, okay? Basically here on the left panels, you'll see this basically log time by second, and also this log size, the different spatial temporal resolutions. And also this is basically the non-invasive and the invasive ones, okay? And then for invasive ones, usually, you know, you only allow you to acquire the data in the patients. But for the non-invasive ones, usually you can do the, you know, for ordinary people. And you know, controls and, you know. And this is basically you'll see that the different degree of invasiveness. And in general, you think about we just use a different devices to measure the spatial temporal information in the brain, okay? And at a very, you know, different resolutions. And actually, when you have the information across different technologies, and actually the signals just are not basically isolated from each other, you know, even though I measure at different scales, but they are related to each other. There's a layer of overlapping models. And this must be testable against the different phenomena. And then these days, typically like a brain initiative, they are very interested in like the several factors. One is basically environment, okay? And also the behavior, I mean, like the human behavior or like a rodent's behavior. Another thing is the neural activities. How do all these different things, the three level of information combined together, basically to integrate and kinds of all the information together to infer about the brain, you know, structure and the functions. And this is a very important way. And these days, this is one of the major directions in the field, basically, how to really integrate information across different scales. Okay. And the imaging processing analysis method, and this is also very important. I think one thing is at the beginning of my career, you know, I was not trained as an imaging, you know, statistician at all. You know, I used to do some other things, but I happened to working in the imaging statistics, and I find that one of the most important things for statisticians, my personal view is, if you want to really understand the basic make a fundamental contribution in the field, I think the most important thing is to know how to process the imaging data by yourself. And that's very important, okay? And that's the reason these days I shift most of my effort to low-level imaging analysis tasks instead of a traditional biostatistical statistical approach. And when you try to analyze the data from the neuroimaging and from different technologies, and you need to realize there are the four common themes in the imaging technologies, the data. The first one, the object. You know that when you analyze the data, you know that your object is very complex. That's the brain. That's the most complex the organ in your body. Okay. And there's a lot of structures and function inside. That's basically they are mingled together and basically function together. Okay. And the second thing is for all these neuroimaging technologies, they have very complex spatial temporal structure inside. Because even though you're using different technologies, then the different technology only take the data at the different scales. Okay, and therefore that's related to for specific technologies, what are the exactly spatial and temporal resolutions, and also in the data because they have the stronger spatial temporal smoothness inside. Okay, because this is not a purely Gaussian noise. Okay. And that's impossible because you know that even so for organ like the brain, this is highly have a very strong structure inside, and also spatial temporal correlations inside. And also this is extremely high dimension because you know that the brain is so complicated. Okay, and everyone is different, and actually they have all kinds of variation inside, but the basic structure is very similar. Okay, and then you have the very similar structures for the brain, regardless you're working on the structure and the function and data. And the first features is basic heterogeneity. And you will ask me what are the most challenging problems when you're analyzing this imaging data and neuroimaging data. Besides the complexity of the objects, most important things when you try to link the imagings with individual subjects and with or without basically the disease, and there's a substantial heterogeneity within individual subjects across the centers and also across the studies, and actually that's the variation could be pretty large in practice when you try to analyze the data. That's very important. You try to correct it. And I think the most important thing is when you have the imaging data, there's pictures, okay? And regardless it's a 3D, 4D, or even 5D or whatever it is. And the imaging data is essentially a function, okay? And because they measure the in grids. And it's basically there are several components are pretty important. One is the device, okay? The different devices, even same brand, they're not quite the same subject, the result may be different, okay? But the variation should be relatively low, and the acquisitions, because there's even the device, they have a different parameters you have to tune, right? And also the data have a lot of noise and different type of noise. You really need to understand what's the key noise sources, and when you do all these imaging analysis, and you need to try to minimize the influence of this noise component to your data. And besides that, and this is the key important part, is basically the brain. Brain is a function of age, genes, race, disease, and other factors, okay? Even like health factors, the way you drink, the type of the alcohol you drink, and also whether you smoke, all the things could influence the brain. Or your astronauts, you stay in outside space, no longer your brain will change. And actually imaging is a function of all the things. But you know that what we care about we care about this red part. Okay, we care about how does the brain changes. Okay, I will care about how the gene influences the brain structures and the function, and also care about the race differences and the disease. How does the disease influence all the things? And this is basically the most ideally generic basically models for you to understand the data. Okay. And it's not just a simple models. Basically, all these existing methods, okay, recover all these aspects. Is basically try to, try to pinpoint basically some key factors associated with the brain, okay. And that's the most important part. And in practice, when you have this type of data, you want to basically develop the method and to analyze it. And statistics is not just about doing and some mixed effect models. Actually, statistics does cover everything we taught for all the methods in the imaging processing analysis method. In general, for imaging analysis, there are two major methods. One is deconvolution, and another one is structured learning. And for deconvolution, essentially, statistics is more like non-parametric or parametric models. And basically, you do imaging reconstruction, and also you do imaging enhance the process. Okay, and it's also a denoising method. Okay, if you think about that's both all come from statistics and also structured learning. And the imaging segmentation and the imaging regression. There are two major methods. Okay, this is also important. It's more like a clustering and also do alignment. Okay, and for most of the imaging reconstruction process, when you started to learn how to process one specific type of imaging data, you need to really go inside to understand some background information and figure out all the details. And this is, I show you one of the very popular equations, for the differential weighted images. And this is also very important, basically, problems in statistics, okay? And basically the left side, you observe the SQ and S0s, and the P is the unknown probability functions, okay? And you wanted to recover this basic probability function. This is the E basically that's related to the Fourier basis. You can choose a different Q and R and basically help you to recover this the P. Okay, and this is typically a non-parametric inverse problems. And regardless you're working statistically and also from the PNAS, that's the problem people are trying to solve. And then actually there's a lot of denoising problems inside. Okay. And also imaging enhance the process, basically that including denoising, surface resolution, Bayes filter corrections, and harmonization. I just show you some pictures about, and the left side is basically the Bayes filter corrections, because there's some low frequency noise drift in the data. You need to remove it to do these Bayes filter corrections. You also need to do harmonization across the studies and across the scanners. And also this is the imaging segmentation type process. If you ask me what are the most important tasks, okay, for imaging analysis, and if you think about the first two, know about statistics, basically it's non-parametric method. That's many people are jumping inside the first things just doing that. But actually for all the imaging analysis, the most difficult ones is the imaging segmentations, okay? And this can be used for many different tasks, and including the quantification of the brain development, localization of pathologies, surgical plannings, imaging guided interventions, and the computer-aided detections and diagnosis in the brain populations. And for clinical purposes, and the surgical planning, okay, localizing pathology, the imaging guided interventions, and computer-aided detection and diagnosis. That's when usually you go to the hospital, and basically you need to pay the price for that. You need to pay the money for these kinds of services. Okay, and the brain fascination is mainly for this research purposes, but then mainly basically how to display. There's more alternate things. You need to try to display the method results on these kinds of brain atlas. Okay, these are all the very important topics. And also imaging regression is another set of the methods. And but in generally, for me, the imaging segmentation is the most difficult, the most important part. Imaging registrations, that's many include the automatic imaging segmentations and the construction brain atlas, localizing the pathologies, multimodal fusions, population analysis, quantification, brain development, and step analysis. And there's another set of imaging kinds of analysis methods. For this type of method, it's also, if you think about this from statistical perspective, it's more like a calculate like a mean and the variance, and also calculate this type of thing. But it is more complex than that. And because usually we do the 4D or 3D registrations on this type of the data. Okay, but I think this is true most of the, if you claim you're expert in either segmentation and the registration, you have to, I think that's very important for you to carry out all kinds of image analysis. Okay, and this is the one of the my students dissertations about basically try to build and atlas, but try to link the function and the structure together. Okay. And this is our recent papers. And in JASA, if you got a chance, you can read it. We try to use the shape analysis tools basically to quantify the changes of the brain shapes across the time. And basically, you can see that how do we do these kinds of analysis on that? Okay, the shape analysis. And what are the most important challenges? It's not about, the key challenges is there's no public available high quality neuroimaging database with detailed annotation information that cover larger spectrum segmentation text and neuroimaging research. Okay, nowadays we have a lot of data these days, but the issue is it still goes back to how do you validate all the tools? And how much you can do about it, okay? And how do you validate all the methods developed in what sense, okay? These are very important, issues in practice, okay? And the larger scale neuroimaging related studies. And actually these days, when I started my career in 2003, and actually that's another, during that time, is usually most studies about less than 100 subjects. But these days they have too much larger scale neuroimaging related studies, and they connect all kinds of the imaging data, not just the neuroimaging, and they can all kinds of imaging data. Even TCGA, you have these kinds of the organ data, okay. And also, this is all kinds of the imaging data. Also, this like this NRST, they have a lung cancer data, non-CT data set. And if you're interested, you can analyze it. There are all kinds of the data, okay, and in practice nowadays. And actually, my personally, spend a lot of time to analyze several data sets. One is ADNI, and the reason I like ADNI because you started from 2004. You have been ADNI 1 to go. And 3, okay, and it's a very long. It's a longitudinal data. It's very important if you're really interested to see this because they can now they try to measure different phenotypes, okay, different phenotypes, okay, biomarkers, A beta tau, the brain structures, and also memory and the clinical functions, and then they try to follow these subjects, okay. And longitudinally for many years, and see when they developed these kinds of Alzheimer or dementias, and also how does the different subjects switch between the different stage. And this data is quite important, and they still are very valuable for carry out a lot of analysis. If you are new to this field, I think ADNI is the first data set that you should try because this is pretty easy to get access to it. And also, there are many people already done a lot on this topic, but there's not many deep analysis hasn't been done. It's mainly because these data sets become more complex. And then when you ask the deep scientific questions, and you need really to integrate the different information together and how to do it. And it's not an easy job because that part is still open questions. And actually, another very important data is the Human Connectome Project and beyond. And the Brain Initiative has been for years. And basically, the Human Connectome Project is a very pioneer project because the primary goal is to build a lot of organ mapping that share like anatomical and functional connectivity within the healthy, the brain, human brains. Okay, and this is basically the reason. One big achievement for this HCP project is basically develop the device, develop these kinds of the basic acquisition kinds of the protocols basically to acquire high resolution structure and the functional images, okay? And actually these days, the NIH invests tons of money basically doing the healthy adult connectome and the lifespan the connectome data and the connectome related to the human diseases. The key reason is they wanted to define what is called the brain development across time, okay? The normal brain development. That's the reason they have lifespan connectors. They want to start from even preterms until for the later stage, like the 90s. Okay, they want to see how the brain developed across the times. Okay, and also they wanted to map this stuff relative in some major brain-related disorders, like neurodegenerative disorders, and in particular Alzheimer's disease, and also neuropsychiatric disease. And these are the very important things. And the ABCD is kind of the part of this kind of effort. And basically, but this is focused also on longitudinal data starting from 2015. And this is brain development and children, health in the United States. And about more than, about 12,000 children aged from 19 to 10. And from the 21 research site, they are going to follow this subject for 10 years, okay? Because this is a critical period, in particular for schizophrenia, okay? And they want to see how they collect all kinds of information and behind it. And actually, another very important data set is these days is UCA Biobank, okay? They collect all kinds of imaging, genetics, health linkage, biomarkers, activity monitors, online questions, repeated baseline assessment and samples. And if you have the resources, or you can work with, collaborate with other people, this is one of the largest medical health related to the data set in the world. And you have to understand, these days, because many groups analyze the data and publish it in top journals, and also all kinds of papers. Every year they publish thousands of papers on this topic. But because this data set is so complex, and because they cover all kinds of domains, it's so difficult to really to dig inside to fully analyze all the different data together, because there's tons of effort behind it. You need to invest substantial effort for fully doing some deeper science on this. But this will give you an opportunity to do it, because we don't have the data. This is one of the best data set in the world, and you just need to access it and try to answer different scientific questions. That's what we are doing these days. And but the problems, there's a lot of things you need to do in order to get the data, access to the data. You need to pay. I don't know how much you pay these days, but usually it's pretty cheap. It's $10,000, something like that. Okay. But this is one of the best data sets, I have ever analyzed. And also another thing is come from my friend ENIGMA, Paul Thompson, basically organized ENIGMA from 2009 to now. And basically you try to work with over the 1,400 scientists across the 43 countries, try to integrate basically imaging, genomics, neurology, psychology, and basically his ambition is a pretty big study. He tried to study a range of large scale human brain studies, integrate data across the imaging, genetics, and many patient population worldwide. Okay, and this is and you have too much data set. At the beginning, you don't have a lot of data set. Nowadays you have too much data set, and the question is, and how to really analyze the data, okay? Because this is need a lot of effort. You have to work with a group of people to work together to do it, okay? It's not easy for you haven't done this type of analysis a lot. Any questions? Okay, and this is the last part. Okay, basically the last part is more related to statistics, and the imaging process is also related to statistics. Okay, and just because, and actually for the when you talk, but the difference is now we are analyzing the image data from a population, and there are 4 additional common themes behind it. One is the sampling bias. Okay, there's a typical problems for all these population studies, and basically you have some sampling bias like under coverage, observer bias, voluntary response bias, survivorship bias, recall bias, and inclusion bias. Those are the kinds of the bias, sampling bias issues when you practice and when you collect the data. And also for most of these large scale population studies, the brain is not the primary outcomes. Okay, this usually is the secondary outcomes. And another important issue is the complex, the missing data patterns. And also this, I know that in statistics, people have been developed methods for missing data problems. But I think that when you're analyzing missing data in the neuroimaging studies, they push you to another levels, okay? Because they are not measure one single, one-dimensional missing data. Okay, now you're missing the whole brain. Okay, and that's the basic missing high-dimensional, even infinite-dimensional basic objects. And how to handle this, high-dimensional objects? That's not it's easy to say, but it's difficult to do. Okay. And there's all kinds of missing data patterns, like missing by design, and like a faulty scanning, attrition in the longitudinal studies, and misentries and non-response in search surveys. And but for this type of typically for missing data problems, in the neuroimaging data is missing by design. Okay? And there's a lot of data studies. You only acquire the imaging data for I only can afford to acquire, 10% of the subjects for the imaging. Then I just choose 10% of subject to do it. Okay. And also for neuroimaging data, there's a complex of data objects. Then you can, because when you have these, the brain is a very complex object. Then we try to extract information from the brain, regardless you're working on function and the structures, you can create all kinds of the complex data objects inside. Okay. The last one is the complicated causal pathways in brain-related disorders. And you know that the imaging basically is trying to measure the structure and the functions of the particular organ, like a brain. Okay. And you can measure this, you can see just like you do the blood pressures. Okay. You can measure your blood pressures across time. Okay. And you can do every day, okay? But the imaging data is the same thing. You measure your brain structure and functions across times, okay? And you think about this, the process is related to, like omics data, okay? Many omics data also and change across times. And also there's all kinds of clinical outcomes and behavior data. And the key thing is basically until this like a disease status, how does the disease progression, right? You wanted to link all these things together in what sense, okay? That's the basically one of the key reasons behind it. And for population-based statistical analysis, there's all kinds of methods can be done, okay? Here I only list about 10 of them. And one of the things is study design. That is still one of the fundamental issues you have to deal with. The second one is the statistical parametric mappings. Okay? The third one is object-oriented data analysis. And the fourth one is imputation method. What type of information method can be developed? The fifth one is basically data integration method. And also the dimension reduction methods, imaging genetics, causality research, predictive models. The last one is a knowledge-based method. Okay, these are the 5 major methods, and actually it's quite important for the neuroimaging field. Okay, and if you can get more general, this basically for all these imaging-related research, and these 5 as 10 topics are quite important. And the study design is still the most fundamental issues. You have case control, cross-sectional, cohort, and experiment studies and descriptive studies. And actually, this is and people are questioning about the UK Biobank, but the UK Biobank is a large population-based cohort studies, and many cross-sectional analysis have been carried out based on the baseline information from the UK Biobank. But also people question about whether this is well known for its healthy volunteer selection bias. And the question whether the results we obtain from UK Biobank can be generalizable to general population. And that's the pretty important questions. And also neuroimaging biomarkers are usually secondary outcomes. That's also very important design question when you consider, you try to integrate the neuroimaging data and to carry out the statistical analysis. And the statistical parametric mapping is very similar like a marker array analysis and other omics analysis, very similar. You're basically, you do and you do traditional statistics and do carry out like linear models to get the p-values, and then you'll come up with you have to correct it for multiple comparisons. Okay, this used to be an important topic, but these days it's not that critically important. And you can do the statistical parametric mapping from the Voxel-wise method to functional models. That's what we did, and other group also did. Basically, you don't really do at each location levels. You treat the data functional data. You can also incorporate a spatial temporal information. Then that's the reason we do multi-scale adaptive estimation and inference procedures. You also can correct the multiple comparison by either use FDRF family and use the Gaussian random field theory, or you can use the bootstrap method. Okay, that's basically when you try to analyze. These are more traditional statistical kinds of a lot of methods that have been developed to in this direction. And another thing is, my collaborator, also my mentor, my friends, Steve Marion, emphasized this object-oriented, data analysis. Basically this, and then he basically tried to say we can develop all kinds of parametric, semi-parametric, non-parametric models for these complex objects and how to really analyze such a data set, okay, by using statistical methods. Is if you are really interested in this, I think Hans Mueller and many his students is working on this. And actually, for all these functional data analysis, intrinsically for object-oriented data analysis, you could use the intrinsic method. And in generally there are three major methods. One is called the feature method. That means basically you're doing some transformation and transform the complex object to Euclidean value of the data and then do downstream analysis. Another one is called the extrinsic method. Basically, you ignore this manifold value data is in nonlinear space. You directly apply classical multivariate regression on it. Intrinsic analysis, basically, you treat the data as the object. And then you assume that the data in the nonlinear space, then you have to do a lot of complex calculation behind it. Okay. And whether practically which one is better, it depends on the problem. Okay, but I think the most useful from practical perspective, feature methods are most important ones. Okay, that's the reason we use it a lot. And also we develop all kinds of intrinsic regression models. At the early stage of my career, I spent a lot of time basically develop the method like put the object in the nonlinear space and then do all these operations on it. You needed to define what is the residuals and define linear functions and all these kind of things. And also this will tell you how do we define the residuals, and also using the geometric structures inside and build this kind of thing such as a framework up. And this is the conditional mean kinds of models we generalize to the manifold. That's the early stage of my career. I spent a lot of time doing this stuff, but these days I'm not. I don't do that much. And another thing is that 10 years ago I started to work on imaging genetics because I'm familiar with imaging. Also, I learned something from genetics, and also why do I care about imaging genetics? Because most of the major brain disorders are heritable, complex traits, and disease. Actually, genetics account for about 50 to 70% of AD risk, 75 to 90% of AD risk, and 60 to 85 schizophrenia risk, and 80% of the ASD risk. For all these complex, basically disorders, and actually many genes, environmental factors, and complex function mechanisms play a critical role in all these different disease disorders. But the genetic part, many genes contribute to this disease. Okay? It's not like a single genes, okay, a few genes. These genetic signals are non-sparse and weak. And actually, this is called the polygenic architectures. Basically, genetic signals small but non-zero contributions. And this is basically related to the imaging genetic features related to reproducibility and heritabilities. And basically, you track all kinds of the imaging features. and we calculate the reproducibilities and the habitabilities. You see that among all the different imaging features, some imaging features are pretty highly reproducible, in particular for related to brain structures. Okay, and the brain functions, because the variation is much larger than structures, and you'll see that basically anything related to like a function of mind, this stuff, the habitability is relatively low, and the reproducibility is also relatively low. Okay, and actually, based on all the extracted the imaging features, we correlated with all the genetic markers. We developed these two larger portals. One is the brain imaging genetics larger portals, and one is the heart imaging genetic larger portals. And we aim to build the best and larger basis on the neuroimaging genetics. And this is some of our recent, we published 2 papers in Science and also 2 papers in Nature Genetics, and also we have several other papers. And on this topic, we just correlate imaging features with the genetic markers. And also we share that more than 2,100 neuroimaging traits across the multiple modalities, and then share the summary statistics associated with that. You can download the summary statistics to carry out the downstream analysis. And the key thing is, these days we still go back to looking at and really the like Alzheimer's disease, and we try to link genetics and with all other biomarkers, A-beta, tau, and also this the brain structures and brain functions, and also clinical outcomes. We really wanted to, because if you think about the sleep are more stationary, there's also another set of the omics data is between, and also these have these biomarkers, okay, and also this behavior and the brain structures. I really wanted to understand how does these things link together in what sense. That's that's the one of the things that we pay a lot of attention on this kind of research. And in practice for heterogeneously clinical defined disorders, the endophenotype is basically why people using the imaging markers for these kinds of endophenotype, because this endophenotype is closer to underlying biologies. And also they can increase the power of the genetic research, because many psychiatric diseases and many the neurodegenerative disease is a very complex trait. It's not a simple 0-1 issues, because the disease phenotype is so complex, okay? And also because this endophenotype can increase the statistical powers. Second one being informative about disorder risk. That's the most important part, okay? And also providing the mechanistic connections linked to the genetic variation to the behavior measurements. That's why we do that. And this is some of the initial model. We try to link things together, like outcome generating models and exposure generated models. This is basically generalized from the causal literatures. And this is basically the way you analyzing imaging and genetic data together. There's a lot of, the dimension is pretty high that we have to develop some of these variable selection tools. And this is the marginal screening tools, and also join the searching screening tools for doing that, okay? And also you can do estimation on that. And besides you're doing this, another thing is basically when you have all these imaging data, also genetic data, and one thing is, and you can construct the gene level, the PNAS, okay, by leveraging the gene impressions, reference panels, and do TWAS analysis. And recently we carry out a comprehensive basic TWAS analysis for all the imaging traits, okay? And to see how whether this kind of information is useful and for disease predictions and other purposes, okay? And this is basically the causal pathways of the imaging, the imaging clinical pathways. And actually we try to link and basic genetics, genomics, and imaging, and also cognition, behavior, and the disease, and also environmental variables, all the different things together. And we know that there's a lot of unseen confounders, but we just use UCAP bank data set to try to build this up okay, and see how does all the different factors link it together, and how do they work together. We explore all kinds of models to answer some clinical questions. Okay. And this is basically one thing that I have been doing for years. Basically, is to try to link genes to the brain disorders. Okay. And the neuroimaging is seated in the middle, and on the right side, this is brain structures, the circuits, and physiology. And we really wanted to go from the genes, molecules, brain cells, the structure functions to eventually arrive at these brain disorders. And also this is confounded with the environmental, social, and the psychological factors. I really wanted to see how does these different information and things sequentially basically happened, temporarily happened, and also how the information link with each other. And this had been in my mind for years, but we made some progress recently, but not that much, because we really needed the data across in each of these blocks and try to link all the things together. And what are the major challenges? And also the complexity of this large scale neuroimaging related data set is too high for most of the research teams in both academia and the industries. Okay. And in many cases, you are good at one data type or two data types, but it's very difficult to really understand all the different data types. It's very difficult to appropriately process the data across different domains with high quality. And that's the most difficult part, and well controlling for potential bias introduced during the pre-processing stages. And it remains uncertain as to how to appropriately integrate the data across the different domains, obtained from different studies, cohorts with possible different study designs from bias data integration. The final one is remain unclear how to appropriately effectively analyze neuroimaging related data sets with the multiple of these to ensure the algorithmic fairness. And if you're interested, you can read my review papers. And you can send an email to me. I can give you my PDF files. And it's not the final version, but it's almost the final version. This will formally appear in August 2nd, something like that. And this is basically, I acknowledge the funding and just any questions, yeah, welcome to us. Thank you.